Transforming growth factor beta receptors respond to TGF-beta, dimeric proteins, that acts as hormones or local mediators. It consists of two types of receptor kinases: type 1 and type 2. TGF-beta proteins are responsible for influencing cell behaviors, cell proliferation, extracellular matrix production, cell death, tissue repair, and immune regulation. Now, let's understand SMADs. These are a group of intracellular signaling proteins that are an essential component for the transduction of signals from the TGF-beta receptor. These consist of our SMADs, co-SMADs, and I-SMADs. Our SMADs recognize the TGF-beta and are phosphorylated by the receptor. Co-SMADs attach to our SMADs after their phosphorylation and are essential for the translocation of the complex into the nucleus. I-SMADs attach to type 1 TGF-beta receptors and block the binding of our SMADs, thus inhibiting TGF-beta signaling. TGF-beta receptors are cell surface receptors. TGF-beta enters the cell vicinity and binds to the receptor. This binding causes the serine-threonine kinase domains to come closer to each other. This causes the type 2 receptors to activate the type 1 receptors through phosphorylation at serine residues. Once type 1 receptors have been activated, our SMADs attach to the type 2 receptors. This results in the phosphorylation of our SMADs by type 1 receptors, causing conformational changes that activate the SMADs. Another R-SMAD is activated similarly. A co-SMAD enters the signaling pathway. It binds to the dimer of phosphorylated R-SMAD. This trimeric unit is translocated into the nucleus to activate the transcription machinery. The trimeric unit binds to the TGF-beta responsive cis regulatory sequence. This recruits the basal transcription machinery. mRNA is transcribed from the gene sequence. This mRNA moves out of the nucleus and into the cytosol, where ribosomes attach to the strand, initiating translation of the required proteins, which may either be used inside or outside the cell. Now let us talk about the termination of the TGF-beta signaling. In order to terminate the signaling, an I-SMAD called SMAD7 approaches the receptor. SMAD7 interacts directly with the activated TGF-beta type 1 receptor. Once bound, SMAD7 helps recruit SMF1 or SMF2, which are ubiquitin ligases. The receptor is ubiquitinated at the type 1 receptor. The I-SMAD is removed. This chain of reactions lead to the internalization of the receptor. Once internalized, ubiquitin molecules are removed by the help of ubiquitinating enzymes. This vesicle containing the receptor then combines with lysosomes in the cell. The lysosomal enzymes degrade the receptor and the ligand, thus terminating the signaling.